This mini PowerPoint will show you how to write up formal lab reports for BIO205. In Blackboard, we're going to be looking under the Lab Materials tab at Labs Lab Reports. Once inside, click on Formal Lab Report FLR Instructions and Submissions. This is where we'll find our instructions, our submission forms, and a sample formal lab report for the gram stain. Throughout the semester, you are responsible for submitting 4 lab reports, each worth 10 points. You choose which 4 lab reports to report on, but they all must be typed in Times New Roman, size 12 typeface, double-spaced, and free of spelling, grammar, and punctuation errors. Reports must also be in your own words, no quoting, as with your other class assignments. Generally, these lab reports are due about 2 weeks after lab results have been discussed in class. The dates are posted below, and we'll go over them in a moment. Basically, each report is to be written to another student that will be able to use your report to understand why they did the lab, what steps to complete, and what results to look for. It will have an introduction, a materials and methods section, results, analysis, and conclusion, and some labs may have a hypothesis, but as you can see below in red, hypotheses are only for specific labs. So we'll start with the introduction. The goal of the introduction is to simply introduce and summarize the lab. What exactly are we trying to do? For example, in the gram staining lab, our goal was to see which type of bacterial cell had thick and thin peptidoglycan. That's something you'd put in the introduction. It's not for results, it's for background, any information that the student needs to know in order to complete the lab and understand why they did it. The second section, hypotheses, is only going to be used in certain labs, and I will let you know which labs in class, and they're notated on here as well. This is basically what questions you're going to answer, and what do you hypothesize about the experiment. For example, in a future lab, you'll be identifying what growth pattern you believe will happen after we use different pHs in tubes, so you will have a hypothesis. But again, for the first few labs, there is no hypothesis. Most of these hypotheses lab will be after spring break. The next section is materials and methods. This is one of the easiest sections. This is where your procedures will be listed in clear steps. Each step should be numbered and expressed in a complete sentence. And in general, methods are usually written in the past tense. Materials should include anything you use, including the microincinerator, any bacterial samples, loupes, needles, or media. After this comes the results. This is where you're going to talk about what you found. But don't get this confused with explaining your results. The results section is simply for you to have diagrams, photos, tables, or graphs, which you can refer to in your analysis section. In this section, you can simply post a picture, like one of the ones you've taken during lab, and write whether it was positive or negative, or what color it was. But again, you should not be discussing any data in this section. Next, you have analysis. This is usually the longest section. This is where you analyze and discuss the results relative to what you found. In some cases, this could be simple color changes, and you explain why this happened. In other cases, you may have to explain whether your hypothesis was correct or incorrect. And then finally, your conclusion. This is where you describe how the results answer your question. It can describe the skills you've learned, the information learned, or some future applications to real-life situations. This is also the section where you can put experimental errors, their possible effects, and ways to reduce errors. This is because in any experiment, since you're all new to microbiology, there are going to be certain errors. Overheating things, bending or breaking things, or media that's contaminated. So you should be honest when discussing these. At the end, if you used any type of material to write up your lab report, which in most cases will be PowerPoints or your lab manual, you will have APA format references for any resources. So make sure you look at the examples below in case you need to cite something. Now before we go on to dates, if you look down at the bottom of this tab, you'll see the example formal lab report for the gram stain, which we'll look at in a moment, and also your FLR submission forms. These look just like your case study and other Blackboard submission forms, and you will attach your document to them and they will be checked for plagiarism. So up here at the beginning I said that you would be doing 4 lab reports, each worth 10 points. But 2 of them have to be starred and 2 of them can be unstarred. So what does that mean? A lab without a star, like Lab 18, motility determination, is a very simple lab. This means that usually they can be discussed in 1 to 3 pages very simply, double-spaced. A lab with a star, like Lab 39, oxidation and fermentation reactions, is a little bit more complicated. They require more results and more analysis. So they're usually anywhere from 4 to 6 pages double-spaced with pictures and references. Now because of all the snow days this semester, we have shifted these dates. So your first set of lab reports, if you choose to do any of these 3, are due by 3/17. So you could easily do motility determination, a very easy lab, acid fast staining, another simple lab, and oxidation and fermentation by the 3/17, and then only have 1 starred lab left to do. Similarly, if you decided to wait till the mid or end of the semester, you could find another set of labs like multiple test media, antibiotic sensitivity, and any other 2 labs to complete your 4. But do be cautious. If you wait till the end of the semester and you haven't turned in any labs, as you can see, you're only left with starred labs, like Lab 34, 35, and 36. So if you wait till the last minute, you'll be doing more work, and in some cases, if you don't turn all your labs in by 5/12, I will no longer grade them. So make sure everything is in by those associated due dates. If a lab must be turned in late, please email me and let me know as soon as possible. But again, these are up to you. You are responsible for knowing which labs you choose and what date they are due. Now down here we have your example formal lab report for the gram stain. When you open this is actually a student paper from last semester. You will see how a student described the gram stain. As you can see, they have an introduction, short and sweet, explaining what the experiment is, why they did it, and what they hope to learn. This particular lab does not have a hypothesis. Then a materials and methods section. The materials are listed, including the cultures that they're using. And because this is a stain, they start after they've created a smear. Generally, when talking about staining, you do not have to tell me how you made the smear, only how you did the stain. So after the clear listed steps in sentence form, we go through results and analysis. Some students choose to make this 1 section instead of 2. That is fine. As you can see, the student has the results in Word format, but also in picture format. Make sure if these are your photos, that you write that. If they're photos from online, you cite them accurately. Finally, we have our conclusion, where you talk about what you learned, any errors that you had, and your references. So like I said, most of these are going to be short and sweet. As long as someone could complete the experiment by reading your notes, most people do very, very well and get the 10 points. But for the ones with the star, they are a little bit more complicated, so expect to do a little bit more work. Start them early. If you have any questions, let me know. And again, make sure when formatting names that you keep them italicized and you capitalize the first letter of the genus. Species is always lowercase. Right, I hope this helps you guys, and I will see you in class.